I'm here today. It's me, Josiah, and I'm back for Bioengineering 102. This is going to be our last class. I know it was a really difficult class. Sorry. All right. I'm here today. There's my voice. It's me. Getting feedback. Sorry about that. Anyway, I'm here to answer your questions today. Come at me with questions. Let me know how things are going. Yeah. It was great. AAV. What was great? The class? Seeing my face? Are you really excited? Was that great? I hope that's what you're talking about. My haircut? I know. You're talking about Hell or High Watermelon. It is great. I'm hoping to get a sponsorship deal. Hell or High Watermelon. Biohacker's choice in beer. Yeah. Come on, come on. What do I think about prime editing? Okay, prime editing. So prime editing is that new technique where they attach a ton of shit to Cas9 and they make it so it increases the homology, repair efficiency, and all this other stuff. First off, they come out with these things every month. They come out with something that's like, ooh, look, there's a better Cas9 genetic engineering thing. And none of them have really caught on because they're not that much better, or they have some problems that people overlook. Now, one of the big problems with prime editing, hey Kent, what's up, man? Hope things are going well. It's shitty about what's going on with GenScript, trying to order your own DNA. Glad you're figuring it out, though. Anyway, back to prime editing. Prime editing, because you attach all these things to Cas9 and stuff, it creates this huge thing that you need. It's over 200 kilodaltons. Now you're talking about this huge genetic thing, or a huge protein RNA complex that can't be delivered easily to cells, right? So normally Cas9 is about, it's much smaller. And most of the time, the place you're going to use Cas9 and homology directed repair is in cells where you can select for the change, right? So you're not really going to use homology directed repair in the human body because the efficiency is so low, so low that the efficiency is so low that it just won't work well inside the human body. So it's mostly used outside the human body in cells, right? So it's good. It might work, prime editing, but it's probably not going to be used that much, and it's not that much better, honestly. It's just that all these people who are doing all this research, they own stock in all these CRISPR companies, and if they do this, if they publish these papers, the stock of the CRISPR companies go up. And so it's I don't know. I don't know what to think. So I would just say, whatever. What companies would you recommend to purchase primers? IDT generally won't ship to people's homes, Tom. So if Tom, with if you're working to have stuff shipped to your home, companies I would recommend are um, not GenScript, GeneWiz, GeneWiz.com, GeneWiz.com. Um, they ship to most anybody, um, and they ship to your home, and they're relatively inexpensive. I mean, primers generally cost the same from anybody, so it's really not going to be that much more expensive depending on who you order from. But IDT, they, you know, they've canceled many orders. I stopped ordering from them because they just didn't want to ship to me no matter what I did. Could you PCR up after linearization of plasmid with endonucleases to get homology again for ligase? Yeah, so generally, what you can do something like that. So you heat up the endonucleases and it should kill most of them. And when you have linearized plasmid, you could amplify, or you don't even need linearized plasmid. You can just amplify the plasmid that you want, um, and generate a lot of PCR product if you're doing PCR. Uh, that's totally possible and fine and reasonable to do. Kent, are there any particular characteristics of an order that might make them hesitant to synthesize? I don't know. You know, it's weird that you had trouble with GenScript because, like, I have them shipped to my, home, and, like, I use my gmail.com email address. So, and they don't care. What I would suggest is always, if you're ordering from somebody, call yourself Dr. So and so. If you can, um, get an email address that's from, like, a company website, and just put up a placeholder website, you know, even if it's just one page. Um, but yeah, calling yourself doctor or whatever, it's not illegal to call yourself um, a PhD. It's misleading, sure, but it's not illegal at all. And that's, if that's what it takes to order some DNA that's not harmful, then that's what it takes. I can't believe they didn't send it to you. Like I said, I've never had any issues with GenScript ever. Yeah, I don't know what flagged them. It might have just been somebody who was, having a bad day or something. You don't have Netflix. How do you see the documentary? I don't think there's any way to see it without Netflix. I think you could get a free trial Netflix account for like a week. Netflix.com. Yeah, I think if you don't want to sign up, or you could pay $15 for 1 month of access. I don't know if you should do that though, because you'll just keep paying because Netflix is good. Do we ship our products to France? Yes, we ship our products to France. That is possible. NAB, yeah, NAB. I've had a lot of trouble. It's so funny because a lot of people I know have trouble with NAB, but again, I don't have trouble with NAB. Again, what I would suggest is create a company, just a website and a company name and a company email address, and like, all these people are stuck in the Stone Age. They think that only companies are ordering this stuff. Why would an individual want to order this stuff? That's crazy. So when an individual orders it, everybody freaks out, even though it's like it doesn't matter. So just create a fake company. SnapVolt. Are we going to do the genetics class again? The genetics class was a tough one. PCR is a lot more error prone than we imagined. And so people have been having trouble with it. If you're interested in taking the genetics class or something like that, email us and we could probably hook you up with the primers and all the stuff that we have for the genetics class. Um, it's just that, yeah, a lot of times with this biology stuff, you try to set it up so that it works every time, and it doesn't, or works as often as possible, and it doesn't. And that sucks, but it is what it is. Um, so, and some things work better than others. Some experiments, some classes work better than others. Yeah. Yeah, no, I mean, if you have, uh, you know, if you want to barcode stuff and stuff like that, let us know. Email us, email me, and what I can do is I can, send you the primers, figure out, get you access, to the class tutorials and stuff like that. Um, but yeah, it's been a tough class, but stuff has worked out. But yeah, it's been a tough class for sure. All right. What else? Any other questions? Any other questions? What's going on? What's going on in your guys' world? We've been really busy. Of course, we're always busy. Um, oh, cool. You got some stream water? Trying to genotype some stuff? Sounds fun. Yeah. Yeah, figuring genotyping can be fun, and it can really contribute to scientific knowledge. Because even like a lot of things that we think are a certain species, there might be a subspecies, or they might even be a completely different species according to the genetics. Um, and so by doing sequencing, you can contribute a lot. You can upload it to the NCBI and stuff like that, and yeah, it's very helpful to scientists. Do I know useful books about bioengineering? There's not really using an iris between multiple sequences. So generally, I think people use can a 2A peptide um, between genes on a mammalian plasmid. Um, I would look at the 2A peptide. It's really small. I've never seen anybody use more than 2 genes in a row on a mammalian plasmid. Um, yeah. How many genes are you trying to use on this plasmid? I mean, an iris will work also, I'm sure. But I think people use a 2A peptide and it's a little bit easier. You got an amoeba eating your rotifer eggs. Hey, man. Life's hard. But good you could find it out using science. Bacteria is generally easier than yeast in transformations. So as you get more complex, so like bacteria is really easy with transformations. Yeast, it's a bit harder. And then mammalian cells, are kind of harder than yeast. And plants and other stuff are really hard. Yeah, Kent, I say for cost effectiveness, the 2A peptide is pretty small. It's only about like 60 base pairs or something. Where do you learn to apply CRISPR to plants? that is, there's no place to learn to apply CRISPR to plants. Um, it's a very complicated thing, alien jacked up. Are you really a jacked up alien, or is that just a fake username? If you use 40, if you use 60 instead of 40, your bands pop out. Yeah, you're probably your concentration of dye is probably more in the 40. I mean, the 60. If you use 60 instead of 40, your bands pop well. Yeah, I don't know. I don't know why that is. Seems interesting. I'll look into it. It really depends on the size and stuff like that. Cannabis strain. Yeah, I know. Everybody wants to genetically modify cannabis. It's but genetically modifying plants is not so easy. And so you know, there's no easy way to do it. It's either going to cost you a lot of money or a lot of time. You choose. Probably both. Probably a lot of time and a lot of money. All right. Keep the questions coming. Might just stare off into space. Forget what's going on. Nice. Congrats, AAV. Your latest mini prep got a two to three times higher concentration. Yeah, man. I mean, people don't understand that. Like science, it's about like the little things. Um, so like a lot of people think it's just like um, oh, I just follow this protocol. All I have to do is read and learn about this. No, it's like, learning how to pipette and do all this stuff, learning what to look for, learning how to know things are going right and how things are going wrong, and when you need to make modifications and when you don't need to make modifications, and all this stuff. Like that intuition just comes with experience and time. And like it's usually what happens in science is like as you spend more time doing it, your intuition gets better, and then your work becomes more successful. It's just the way it is. Do human cells have resistance, or do they depend on our general body's defenses? Um, so if you're talking about like human cell culture, so generally normal human cells don't really have, if you're talking about like antibody defenses and stuff like that. Um, no, generally those things come from like T cells. So normal human cells, if you're doing human cell culture, they won't have any strong immune responses or defenses or anything like that. So you can, like infect them with viruses and other stuff really easily. There's a green hue to it, the wash buffer. I don't know. Why it's got a green hue to it? That sounds interesting. I don't know if it should. Generally, wash buffer I think is just tris and ethanol. Um, like 50 millimolar tris and like 70% ethanol. to a volume of 70% ethanol. Um, I'm pretty sure, but I could be wrong. Look it up. But yeah, I don't know why it's got a green hue to it. That, you know, who knows what that is. Sorry about that. So some mini prep kits, they have like pH indicator dyes and stuff like that. So it could just be something like that. Any recent articles or developments in bioengineering that really grabbed my attention? No, I'm starting to get more interested in the bigger stuff like ecological scale. You know, the mosquitoes and gene drives and stuff like that. Ecological scale stuff is interesting. We can't predict it that well, and it's just an interesting phenomenon. So, Tom says, I've read some literature that says you let agros cool down a bit before adding fluorescent dye. I don't see a difference to your take. I don't wait. And I've never had an issue. I, you know, I think you have to understand, like, science is a practice of, like, being meticulous and anal, because some experiments are very finicky and have a very small chance of working. But most of the experiments we do nowadays have been optimized, right? So it's not that big of a deal, you know? We're not like heating agarose on like a pan over a Bunsen burner or something. So we don't need to let it cool down. I've never had an issue, and I add my dye right after I take it out of the microwave, so shrug. You know, I think just do whatever you're comfortable with. When your science is working, when stuff is working, just keep doing it, right? I think that's the rule of science. It's just like, if stuff is working, why change it? You know? Do what you're comfortable with, because that's when your science will be the best. Yeah. So, like, not everybody does science the same way. Even biohackers, you know, people who are doing science in their home, like, everybody has, some people are super anal about stuff, and other people like me are just like, whatever, you know. The amount of time I waste doing all this stuff, I can just redo it when stuff goes wrong. And yeah, generally it works out. I think that science, though, in general, is like getting an intuition again for like what you can change and modify and what you can skip and not skip. Just like it's okay to challenge the status quo, to challenge what academic scientists do. They try to say there's a reason, but a lot of times there's no reason that they do stuff a certain way. Alien jacked up. Look, we're not talking about plants here. Move on. I haven't tried gold instead of platinum. Honestly, I don't know what would happen to the gold. The reason you use the platinum is because it's pretty inert, right? And so it doesn't get electrolyzed. I don't know if the same is true for gold. So if you're going to try gold electrodes, you know, it's up to you. I'm sure somebody might have tried it out there a long time ago. You should look into it. Platinum is pretty expensive, but yeah. What you gonna do? Yeah, give it a go. Let us know how it goes. You know, I'd be interested to hear. What else? What crazy experiments are y'all dreaming up besides plants, engineering cannabis? You know, the problem with engineering plants and stuff is nobody has really ever given me like a moving pitch, you know? Barry, rewind to the beginning. We already talked about prime editing a little bit. Check it out. Is doing gene drives or breeding dogs really hard? Is it hard to germline edit harder germline edit than somatic edit? So really the main difference between germline editing and somatic editing is just that like you're working with embryos and cells instead of with a big human, right? If you inject DNA into your arm or your muscle, um, like there's a lot of cells there. So there's a lot of things that the cells can get into to edit, right? If you're working with an embryo, it's a lot more difficult because you're working with very few cells. So generally, you're doing some sort of micro injection or something like that, um, and the cells. Oh, sorry about that. The cells or what you're working with are much more limited. So it's easier for stuff to go wrong, right? If you're editing an embryo at like the 8 cell stage or something like that, and you mess up some of the cells, like it's going to mess up the embryo. Whereas if you do a somatic tissue edit and inject an adult muscle, and 1,000 cells die, you're probably going to be totally fine. Your probably body won't even respond, right? Wait, you need a permit to get your hands on C. elegans? Are you in the U.S. AAV or no? Sounds like you're probably not in the U.S. because C. elegans you shouldn't need a permit in the U.S. to work with it. I mean, you can just order it online from many different places. The problem I found with C. elegans, the little worm, Australia, okay. Problem I found with C. elegans is that they're too small. If they were like double the size they are, they would be perfect. Maybe somebody should do that. Engineer C. elegans that's like twice as big. It just needs to be twice as big. That's it. You know? If C. elegans is just twice as big, then it'd be a great organism to work with. But it's so small. Yeah, one day I want to do, embryo editing. You could find microinjectors and stuff on eBay. Um, it's not that difficult. You could get yourself set up for pretty inexpensively. So it would be fun and interesting. Um, I don't know if it would be very scalable to get other people to get involved because getting microinjectors for everybody for a class would be quite complicated. But, it still be something that would be really fun to learn. Yeah. Hmm. Yep, exactly. Hey man, I'm not doing anything illegal, that's the thing. Microflora to express Komodo dragon antimicrobial peptides. First, I would just start off with normal lab strains of E. coli, Kent, if you're trying to express some peptides. That's the best thing to do is start off with lab strain of E. coli, so you have a very controlled environment to do it. Um, After that, honestly, the toughest part about finding good flora on your body is finding stuff that isn't necessarily antibiotic resistant. Like a lot of the stuff on our bodies are naturally antibiotic resistant. So, take a skin swab of yourself and streak it out on like a kanamycin or ampicillin plate, and you'll get growth. So, first, what you have to do is you generally have to select for strains that don't have antibiotic resistance. So, swab your skin, grow it up on some LB, and then take colonies from the LB and try to streak them out on different antibiotics, um, and see what's killed by the antibiotics. Uh, because the easiest way to do some modification, genetic engineering, is through antibiotic selection. Otherwise, you're just going to have you know, a interesting time. So figure out the antibiotic selection first, and then go with that. And then you could, do 16 S sequencing. If you got a PCR machine, um, you know, 16S PCR amplify the strain you have, send it off to GeneWiz, um, blast it, figure out which strains it is. Most likely, it's probably going to be, I think, what is it? Really popular on the skin: Staphylococcus, Streptococcus, E. coli, Klebsiella, um, Bacillus, stuff like that. Maybe, hopefully. Awesome. Got a PCR machine. You're ready to go. Ready to rock it. Identify some of those bacterias. Komodo dragon antimicrobials. Kent, you should start a blog or something. Start keeping track of your experiments. What influence will CRISPR-Cas9 have in the future? I think there's a lot of hype behind CRISPR. CRISPR is really great for like science experiments and stuff like that. But in terms of like clinical medicine, I don't think CRISPR-Cas9 will ever be used. Oh gosh, did we lose stream? What happened? Uh, Seems like my camera went out. Hold on. Hmm. Looks like I'm frozen. What's going on? There we go. All right. Sorry about that. Um, yeah, I don't think CRISPR-Cas9 will ever be used in the clinic. Or if it will, it'll be like the most miserable attempt. Um, like zinc finger nucleases have been around for 30 years, and they were used in the clinic once, and the results were, you know, mediocre to not good. And yeah, the thing is there's not many diseases that you can use CRISPR on. There's not many, because you can't use. So when a lot of people think of CRISPR, they think of changing genes. You can't do that with CRISPR in the clinic. The efficiency is so low, it doesn't work. So really, the only thing you can do with CRISPR in the clinic is knock out genes. So genes that are bad, you can knock them out. The problem is there's very few diseases where you actually want to do that in an adult. There's very few diseases where you just want to knock out the bad gene instead of just replacing it with a copy of the good gene, which is what normal gene therapy does. Normal gene therapy just replaces the copy of the bad gene with the good gene. So, you know, I think CRISPR, its main benefit, you're hearing double of me? Echo? This is just struggling then. I only see one audio for myself on the stream thing. Yeah, I mean, are you guys still getting feedback? Are you hearing? Are you getting hearing double of me? Sounds fine. Kent, here's the problem with gene drives: is gene drives are so slow, right? Joe, you might have two windows open or something, or I don't know. Check and see. Try refreshing. The reason everybody uses mosquitoes with gene drives is because gene drives are so slow. Gene drive has to wait till the animal can procreate, and then in order for it to propagate, it has to wait for the next generation to procreate, right? So if your procreation time frame is longer than if it's longer than like a mosquito, which is like a week, like you're gonna have such a hard time infecting a population, right? If you think about it, right? Say you have 1,000 rats that you release, right? And those rats mate, and they release, you know, double. So some of the rats die, some of the rats emigrate. So let's say it goes to 2,000 or 5,000, right? And what you're constantly doing is you're fighting this battle to get bigger and spread, and your procreation time is like a year, maybe 6 months or something like that. So you're talking by the time you reach a high enough threshold to actually cause population collapse, it's going to be like 20 years, 50 years in rats, right? Or else you have to release like a huge amount. You have to release like half the population. And even then, like you're going to have Like not all of them are gonna mate. Some of them are gonna run off to some other field someplace. Some of them are gonna die. Um, that's what makes it so interesting, though. I think gene drives is not necessarily the gene drive, but like all the reasons that gene drives don't work. That's what makes them so interesting. Is like the famous Jurassic Park, like life finds a way. Life also is just like it doesn't do what we want it to do. In a lab, it's really easy to like lock things in a cage and be like, look, you know, um, like you can cause a gene drive to work. But when it's out in the field, you have stuff like immigration. Mosquitoes immigrate and emigrate, right? So you have new mosquitoes coming in and mosquitoes that have been mutated going out, going to different populations, right? So it's like, it's such a complicated thing that it's very difficult to actually like predict and make to get gene drives to work, especially on stuff that like, funguses, like goddamn, they're slow moving, you know, diploid funguses, I'm sure they don't mate well. They got to be like mixed or right next to each other, or yeah, just be a long drawn out process that while it may eventually work, it's like, so what they do right now with gene drives generally is they keep releasing, right? It's like you release 100,000 mosquitoes and they start the population declining a little bit. And then like a week later you release another 100,000 and you just keep doing this over and over and over again. You release mosquitoes and it's not such a simple process as the popular press and regulation makes it out to be. Even scientists blow it out of proportion. So, have you used prokaryotic and eukaryotic antibiotics like G418 to knock out fungus as well? I've not worked with fungus, so fungus is a little bit more difficult to genetically engineer because they have, the cell walls that are really tough, so you got to break that down. So it would work for sure, but like the GFP plasmid that comes in our yeast kit should technically work in fungus, I think. But I have not spent enough time working on fungus genetic manipulation. And so, but there are some simple papers on it where, the process is just treating the fungus with, I think, it's not amylase, it's another enzyme that'll break down the cell wall so that you can get cells that you can transform. I have not been successful with it though, but I haven't tried that much, so there's that. How about using gene drives on ticks? I mean, again, I don't know much about ticks, tick mating, and all that stuff, but I think it's probably going to be complicated. There's probably a better way. The fungus class is nothing biological. Because it teaches you how to grow mushrooms from like mushrooms that you get at the store and stuff like that, or wherever you get spore prints or mushrooms from. It's pretty cool. We actually have this mushroom farmer who's teaching the class for us, who's recording the videos right now. But yeah, it's no biologicals included, so it should be able to ship all over the world. And it's just basically to teach you how to grow mushrooms. Anything. You like shiitake mushrooms? I do. Teach you how to grow them. Give you the materials to grow it, like all of our classes. Yeah, it's going to be a good one. All right. Any more questions before we close it out? Close it out for the day. Close out the class. I guess we didn't even really talk about 102, but hey, it happens. For people who are in the 102 class, we'll be around. If you want to ask questions, keep working on the class. We're always there to answer your questions and help you out. So keep working on it. Good. Glad you learned something from the class. Um, Hopefully you'll be able to use it to genetically modify some cool things. I hope. Oh man, the human cell culture class is going to be really fun. So hopefully you receive your stuff soon. Did you receive your stuff yet, Kent? Oh yeah, Joe, the class pages will be up indefinitely. So the class pages aren't going anywhere. Don't worry. They'll be up. Um, yeah, I love shiitake also. They're really good. The thing is, Sylvia, actually you don't need much space to grow shiitake mushrooms. Like, you can buy them from the store. This is what I'm learning from this class. I'm also learning from the class from this mushroom farmer. You can buy them from the store, take pieces of them, inoculate, just like cardboard until you start to get spores and other stuff forming. And then you can inoculate a bag of grains, of sterile grains, and the mushrooms will just grow. And it's only something that's like this big. It only takes up like that much room. Just stick it in a cupboard or something, cabinet, and let it grow. It's really small and easy. You only got the microscope so far. Well, hopefully everything else will arrive soon. Um, you got your boxes for 201 today? Awesome. That's exciting, Leslie. Pretty cool. Take care of those human cells when you get them. Treat them well with respect. Yeah, use coffee grounds. Yeah, sorry, we can't send. We're not. I wanted to. Once you start getting to sending human cells around, things start to get a little more sketchy and iffy and non-law abiding. So we're trying to stay away from that for right now. uh, I've been trying to figure out ways also to maybe like dry out human cells or something to make it easier to send. I mean, one of the problems with sending them internationally also is just like um, the time it takes. Overnight shipping internationally is expensive, or like short shipping. If you have though like a university or someplace that you work with, I think I could send the cells, but otherwise the customs and stuff. Awesome, Leslie. Cells are at 37C now. Perfect. Good to hear that. They thought you were a truck lab. Hey, it happens. It happens. All right. I'm going to get going. I enjoyed hanging out with everybody, answering your questions. Um, you know, the Genetics 101 class is tomorrow at 4 p.m. Genetics 103 or whatever. Bioengineering 101 is on Thursday, and then next Monday is the 201 class, which will be really fun, learning to split cells. But yeah, thanks for coming. I will talk to you soon, and we will meet again.